Hello and welcome back to Multibiomics. This is the third part of clustering and annotation. Right, we have already clustered our cells in the second part of our video. So if I tell you the workflow, basically we had reduced the dimensionality of the data set with the help of an algorithm. It is an unsupervised algorithm known as PCA. So the PCA, finds the major variances in the data sets. So we have found two principal components, that is PC1 and PC2. So PC1 and PC2, these are the set of genes which provide the maximum variation in the data set. Now, after that, we had, excuse me, so we had found that with the help of PCA, we could cluster our cells. So if I visualize the PCA here, the dim plot, if I run, okay, so this cluster will get, now here the colors are different, right? Because we had already performed another dimensionality reduction and 9 clusters have been found in the previous video. For that, you can go and you know um, look at the video. I'll be providing the link to that video in the description box below, or you can alternatively click the cards in my video over here. But the color, so the plot was something like this, and there was no like different colored points, but we overall we got 3 clusters there. So this is the first cluster, this one is the second cluster, and third cluster. If you couldn't understand, please go back and look at my video there. So same graph you'll get, but they'll not be colored differently because at that point of time we had not chosen more principal components, and we had not run the U map, that is the non-linear dimensionality reduction. Then we ran the elbow plot to determine what should be the number of principal components that we'll be proceeding with. So here the elbow plot, if you see, uh, each points represent a principal component. So this is the principal component 1. So principal component 1 are the set of those genes which provide the maximum variance, and as the principal components are increasing, the variance is going down and till the point 10, right? So that is principal component 10. Till that, we are getting a fine amount of variances. So how to interpret is that the point from where your line is getting straight, that is the point after which the variance is not much of use. So that is we can carry on with only the first 10 principal components, or the first 10 sets of, not the first 10 genes, but the sets of genes. First 10 sets we'll be picking, and each set will have many genes. So principal component 1 will give you the maximum variance, and the principal component 2 will give you the second most variance with the standard deviation between 4 and 5 somewhere, right? And you see principal component 3 is just below 4. So the standard deviation is decreasing, and standard deviation is a matrix uh, where we can interpret that the variance, how the variance changes, right? So if I run this command and I just look at the 5 cells which I've already done, you see this is the first cells which has been assigned to the cluster 0, that is the first cluster. This is third cluster, this is second cluster, this is first cluster again, and this is assigned to sixth cluster. So what are the levels? How many clusters you can find over here? 9 clusters are generated from 0 to 8. Okay, and if we want, then we had performed a U map, that is a non-linear dimensionality reduction, because now we don't have only 2 but 10 principal components, and according to that it will try to cluster the cells, right? And if you want to visualize the map now, so because of performing this step, now we are getting different colored cells, right? Because each color, so spatially also they have been clustered. I'm getting this cluster, big cluster over here, a small cluster over there. and this cluster over here, and very few cells over here, right here. Okay, but even the points that are grouped together, there may be some subpopulation of the cells which are not same. So those cells are colored differently, and it was possible only because now we had performed a non-linear dimensionality reduction known as UMAP. So UMAP one, you can say, are the set of features that has more variance. You know, the distance between the 1 unit distance between UMAP one gives you the most variance when you compare to the distance 1 unit distance between UMAP two. As I've already explained you in the PCA, the same logic goes over here also, right? So moving on, we will be now moving on with the clustering. Sorry, yes, with the annotation part. We'll be proceeding our work and we will try to find the differentially expressed features, right? The features that are differentially expressed, and they will be serving as our biomarkers for. So the biomarkers are the transcriptomes or the gene expression levels. Okay. So, and with the help of that, we'll be clustering. So find all the markers of cluster 2. For example, so this is the third cluster, which name is cluster 2. So we'll in the cluster 2, what are the differentially expressed genes we'll be finding, and we will be saving it in a variable known as cluster 2 dot markers, and the function that is used is find markers. So Surat gives you this function already. So it will look inside the Surat object PBMC. Okay, and so the identity will be this 1 equals to 2. It denotes which cells the which cluster the cell belongs to. So we are selecting the cluster 2. Okay, and if we look at the, have we run this? No, so we have to run it first, right? So we ran it. Okay, so there are some. So yet assay data is deprecated, right? So these are not an important warning, and we'll be proceeding forward by seeing this. You see, so this is um, the value that we are getting. The gene names, the first 5 gene names we are getting in the cluster 2: LTB, IL32, LDHB, CD3D, and IL7R. So these are the marker genes that we are getting. So in cluster 2 itself, we are getting these many different types. So you can go with the uh, you know um, literature survey, and you can find out that what are these marker genes associated with. Their values are also given. So if you want to interpret this data, let us just look what this data is telling us. This output actually shows genes that are significantly more active in the cluster 2, because we have given the cluster 2 argument. Ident is equal to 2. It will capture all the cells in cluster 2. And it is going to compare the cells in this cluster with all other cells, right? So for example, if I take over here, hmm, so the example is, okay, the first one is LTV genes. There are several columns like p-value, average log2 FC, PCT1, PCT2, adjusted p-value that we are getting. So the first genes that it's getting, that is LTB. So you can interpret this, if you know the meaning of the value. So why is it placed on the top? That means this gene is expressed much higher in the cells of cluster 2, right? And the p values, if you see, the p values are like 1.75. So it's lower p value. The p value which are less, they indicate that it is strong statistical significance, at least like 5%, p values are there, then you can convert these decimal points into percentages, and you can see the smaller the value, it will be like more strongly statistically significant. So likely these genes are the marker genes of the cluster 2, and they will help us to define the unique identity or the function of these groups of cells. So we have to do literature survey, or we can go for the um, uh, online databases for cluster. Those online databases also are available, like DisGenet, biomarker base, OMIM, right? Uh, Biomarkers databases. You can also go for the gene expression and transcriptomics data in websites like Human Protein Atlas, GEO, Expression Atlas. So all these gene related biomarkers, you can go and search there. Also, you can do the literature survey, and you can understand that what are the functions, and with the help of that, we can understand what group of cells these belong to. Cells in the cluster 2. Now, suppose if you want to find all those marker genes which are separating or distinguishing the cluster 5. This is just an example. Suppose you want to now study cluster 5, and how are they different from all the cells in cluster 0 and cluster 3? Now, these things, if you want to compare, so what we are doing, we are assigning a variable cluster 5 dot markers, and again we are using the same function, find markers function in PBMC. So the identity of ident.1 is equal to, now we are taking the fifth cluster, right? And we are identity 2 is the one from which we want to compare the first cluster, right? So the ident 1 is equal to the cluster that we want to compare, and ident 2 are all those clusters with which clusters we want to compare with. So if I run this, I will be getting the data, and we'll be looking at the data for the first 5 lines. And here, these genes are responsible. So FCGR38, IFI-TM3, CFD, CD68, and RP11. So this is the gene that differentiates all the cells in cluster 5 with all those cells in cluster 0 and 3. So like this you can find out, and find the markers for every clusters compared to all remaining cells. So all the combinations we can also do. So that will be storing in pbmc.markers, and the function here that we're using is find all markers. Okay, not find markers but find all markers in pbmc. Okay, and the position is true, so it will give you the positions also. And so pbmc.marker, we will be grouping by the cluster. Clusters are already generated 0 to 8, that is 9 clusters total, and then we'll be filtering it with which column, the which values, the average log to FC value, this value. So we'll be filtering it with the help of these values, right? Okay, so let us just run these. Okay, I'm running it. Okay, so all the clusters are being calculated, and it's a huge data again. 7,014 more rows are present over here. So we have calculated with the base of, all those values we have done. But we'll be filtering with the help of average log to FC values. Now, choosing log to FC values, that is actually log to fold change, right? So whenever you are using, when you want to identify the markers, see, biology is totally different from mathematics, right? So You have to ensure that you focus on the genes with biologically meaningful expression differences, not only the statistically significant ones. Right? The biological relevance plays a very important role. Will get an improved marker quality also here, and the noise will also be reduced if you use the log2 FC, that is fold change values, log2 fold change values. Um, it also gives an advantage. It also gives an advantage of proper visualization. You can make proper volcano plots and heat maps and feature plots, which will be more informative when showing genes with meaningful expression shifts. Okay, and in fact, better downstream functional analysis also can be performed. So gene sets with large fold changes often enrich more clearly in GO or KEG or pathway analysis. Okay, so that is why we have chosen this particular metrics for finding the marker genes. So moving on with our workflow, we can also perform, ROC test to find the marker genes. Let's go on with that. I'm just pasting the code over here. Yeah, because typing will take a long time. So now when coming to using this method, the ROC method, it is known as Receiver Operating Characteristics Test. Now it is a method to find marker genes, and what it does, it measures how well each gene can be distinguished, or how each gene can distinguish this particular cluster, say cluster 0, from all other clusters. So instead of just checking if the expression is different, it also checks how accurately a gene can separate the cell from, cluster 0 from the rest of the clusters. Now it is like a classifier. It gives an area under curve score, AUC curve score, between 0 and 1. The higher score are closer to 1, 1 being the highest score. And the higher the score, it means that the gene is a very strong marker. Mistake those genes as the markers. So this method is useful when you want genes that clearly and reliably define a cluster, and not just statistically, but also in terms of classification power. So here, so cluster 0 dot markers already we had. Okay, we had not made. So we are making on another variable like cluster 0 dot markers, and again we are finding the markers. What we are doing in this, so the first we have to compare the cells in the cluster 0, and we are choosing log fold change value, and we are giving a parameter threshold value over here. And what is the test that we are performing? ROC, right? And later on, we'll be visualizing them with the help of a violin plot. I'm running this ROC. Okay, it will be helping us to find the markers. So calculation is done. Right. Now plotting the violin plot and giving various colors. Right. So you see over here. So expression level of MS4A1 gene and CD79A. So here in this violin plot, how to interpret this one? Um, when you look at this violin plot, it shows the expression level of two genes, and those are like MS4A1 and CD79A. And all the clusters are mentioned over here for each gene, right, from 0 to 8. So interpret that how? So in the left plot in MS4A1, It is highly expressed in the third cluster with almost no expression, very less expression in the other clusters. This suggests that MS4A1 is a strong marker for cluster 3. CD798 in the right plot, it shows a very similar pattern, very high expression in cluster 3 and very low or negligible expression elsewhere. So like MS4A1, it is also a marker gene for cluster 3 only. The orange violin shape, like it is giving, represents the distribution of expression. As you can, as you already know, the width of the violin plot will give you the distribution of the data, or here the expression levels in each cluster. The wider the violin, the more cells will have that expression level. And each dot are the individual cells, right? Now, if you want to give you more information, it's like MS4A1 and CD79A are actually the B cell markers, right? So now these are the B cells. And their strong expression, both of them, in cluster 3 confirms that the cluster 3 is representing B cells. So that is why you have to go for literature survey and the database to find the biomarkers, right? This is the common finding in the PBMC dataset, and it actually supports the accurate cell type annotation. So we are going in the right track. Fine. So moving on, let me close this one. Okay. We can plot raw counts as well. So what we okay, and then feature plot. We'll just what we are doing. We are then making a violin plot. We're making a feature plot, and then we grouping them by clusters, and we'll be drawing a heat map. Yeah, so raw counts are nothing but the unprocessed number of reads, right? Before applying any normalization. So we can, plot that well as well. If I plot this violin plot over here and run this, okay, I'm getting this. Before normalization, anything. So this, we have chosen NKG7 and PF4 gene. So you see in the cluster, in the fourth cluster, the expression level is quite good. And we are also getting it in the fifth cluster, sixth and seventh. And the PF4 genes, we are getting the expression in the eighth cluster, right? And you can see the distribution of the cells. So each point again is the distribution of the cells. So like that, you can go and verify. So raw reads also can give you some insight, but better plots are, better insights you'll get after normalization and standardizing the data, right? So now we'll be generating a feature plot. So you have got some important genes, for example, like MS4A1 and all these genes. You have different combinations of the clusters. You have studied with the help of find marker genes, and you got some genes, and you think that these are significant ones, and then we'll be running this code, this command, and we are making a feature plot. You see, for each gene, we are getting this cluster, right? You see, so MS4A1 has high expression in this cluster. You can go and see your UMAP again, and you can find out what in this cluster, what are these cells with the help of colors, right? So you can compare that plot. All these genes, different genes, are expressed in which type of cells. You can visualize with the help of this feature plot. Again, if I take the top 10 genes. So what we are doing here in this line is we are grouping all the markers by clusters. The clusters are formed and all the markers should be grouped into Sorry for that. All the markers should be. So the pattern of the clusters should not change even when you're clustering it with the help of you know your found biomarkers, because they are the one uh, helping in clustering, right? And here we are keeping only those marker which has strong log 2 fold change values, log 2 FC values, and we are going from that. We'll be picking up the top 10 genes from each clusters, and then we will save into a variable known as top 10. Okay, so we are doing this here. I'm running this particular set of code. Done. And now we will be visualizing that with the help of a heat map. So this heat map is generated, and top 10 genes from each clusters we have taken right now. So each cluster is giving us the top 10 genes. Okay, so this is the top 10 genes of cluster 0. This is the top 10 genes of cluster 1. You see, we're getting a diagonal relationship. Like that we can find. Well, how can I interpret? I can interpret because, uh, you know, the yellow color. Right? The top 10 genes of each clusters, very nice pattern that you can visualize, right? And yeah, there are like yellow scattered here and there, so it doesn't mean that other genes cannot be expressed, but yeah, definite pattern, this diagonal patterns. These are the top 10 genes of cluster 0, then cluster 1, 2, 3, and you see in these genes are co-expressed with these genes. If you can follow my cursor over here, right? All these things you can interpret. There are disturbances, but I hope you don't mind them. We have just finished most of the work and moving on with assigning the cell type identity to the cluster so that we can annotate, right? So there's a variable that we are assigning new cluster IDs and all these genes that we have identified, right? And we will be naming our clusters according to these genes. Now the clusters will not be named from 0 to 9, but it will be named according to these genes, and that's what the annotation means. So in the Surat object PBMC, we will be using the function rename idents, and we will be providing them with the new cluster IDs that we have already saved over here. Okay, and then we will be performing a plot. So like this, you should look at the code and you can understand also. It's very easy. It's not hard, right? So here you are not only learning the workflow, but you are also learning how to code. Even at least like if even not like hard, coding from scratching, at least like if you get a code, if you get a script, you can interpret what's happening there. Right. So if I run this, now you see, now the annotation has been done. The, you know, these are the platelets. These are NK cells. The green, so the green one below are the B cells, and this nearly bluish green color CD8 T cells. Okay, and this yellowish green color over here, little bit of hint of yellow and green. This is memory CD4 cells. These speech cells are knife CD40. The blue cells are the cells, and also these are like FCGRs 3A cells plus monocytes and CD14 and monocytes, right? Then these purple cells are few cells known as dendritic cells. All these things we have already annotated. And so we'll be visualizing some plots. So let us see here, running this. Fine. So UMAP 1, UMAP 2. So we'll be visualizing the UMAP plots again. I'm plotting it after calculating. So we are getting all these clusters again. So we are just adding the names over here. Which color represents to what color? and you can save this. If you want this in the PNG output or JPG output, you can save this whatever height and width of the plot. If you, because if you want to add them in your paper, if you're doing some work, so that you can assign the dimensions over here, right? And you can also save them into your disk. Now these last two steps I'm not doing it. I don't need them also, and I've already performed this several times. But when you perform this, you write this command save RDS. So it is whatever you have done till now in your Surat object, that is saved as it is, so that next time you can just call pvmc3k final.rds, and all these things will already be present in your Surat object, right? So, guys, with this, we are wrapping up our first module, clustering and annotation. Um, you can feel free to go through all the videos at once again, and I'll be coming back with the module 2 uh, in a span of few weeks, right? And I would also encourage you to search for new papers and reproduce their work, and that is the best way of learning. So I hope this module was interesting and you found it insightful. I hope you have a nice day, and thank you for joining me in this journey. And thank you for supporting me. And please like, share this video, and subscribe to my channel. Thank you. Have a nice day.